What if the most devastating genetic diseases could be deleted from a family tree forever? No more cystic fibrosis, no more sickle cell anemia, erased at the very moment life begins. For the first time in history, we aren't just treating the sick, we are editing the blueprint of the unborn. But as we pick up the molecular pen to rewrite the human embryo, we have to ask, where does the healing end, and where does the designing begin? This is the high-stakes world of embryo editing, and it is the most complex codon we've ever tried to crack. Welcome back to my channel. I am the Geneticist. Today in Research Codon, we are stepping into the most controversial frontier of modern biology. We've explored personalized meds and lab-grown tissues, but today we go back to day one. We are looking at the science, the risks, and the deep ethical shadows of editing the human embryo. To edit an embryo, we need a tool that is both a GPS and a pair of scissors. We use CRISPR-Cas9 to find a specific mutation and cut it out. But we are also seeing the rise of base editing, a more surgical approach that doesn't cut the DNA but instead chemically converts one genetic letter into another. While some studies have shown incredibly high correction rates for specific mutations, the process is far from perfect. We are moving from blunt editing to a precision that was unthinkable just a decade ago. Despite our progress, we face a major technical ghost: mosaicism. Sometimes the edit doesn't happen in every cell of the embryo. This creates a mixed population of cells, some corrected, some still carrying the disease. There is also the risk of off-target effects or large unintended deletions. In the delicate world of an embryo, a single mistake in the code can have echoes that last a lifetime. Safety isn't just a goal here. It is an absolute requirement that we haven't fully mastered yet. How do we get these tools into a microscopic cell? Traditionally, we used microinjection, but new innovations like iGonad are changing the game. This method allows us to deliver CRISPR components directly into embryos within the oviduct, removing the need for invasive isolation. We're also seeing AAV-assisted electroporation, which can achieve editing efficiencies of up to 90%. We are making the impossible edit more accessible and scalable every day. But just because we can doesn't mean we should. Embryo editing raises questions that science alone cannot answer. If we can erase disease, will we eventually try to enhance traits like intelligence or height? Could this create a genetic divide where only the wealthy can afford to give their children a biological advantage? Most importantly, how do we get informed consent from a person who hasn't been born yet? We are rewriting the future of our species, one letter at a time. We are the first generation of humans with the power to direct our own evolution. It is a power that requires equal parts brilliance and humility. The research codon of the embryo is the most sacred code we have. If you want to examine the technical papers on mosaicism and the iGonad protocols I used for this episode, the full research is waiting for you on my Telegram channel. Thank you for joining me for this profound journey. Stay curious, keep those cells dividing, and I'll see you in the next decode. See you soon.